more efficient drug pipelining. Carsten, give it a go.
Thank you. Thanks for the wonderful introduction, and also a warm welcome from my side, from sort of the other part of the world, to everyone who's participating today. Thanks for taking the time. Yeah, we'll be talking a little bit about more efficient drug pipelining, and this sounds like a very bold title. Obviously, we can only make a rather small contribution to the efficiency, but this contribution is still pretty important as we think. So we will be showcasing this contribution by the means of our medicinal chemistry software, SeeSAR. And before I want to go into this, I would like to discuss a few bottlenecks in drug discovery. Please bear with me. Obviously, I cherry-picked those a little bit to fit our topic. There's a gazillion bottlenecks out there that make drug discovery a very tedious and long-lasting process. But the first that we oftentimes experience is that the computational and the medicinal chemistry people are too separated, and that may be in a physical but also in a mental sense. So both departments can be quite on their own, although they should be talking to each other or should be more fluently talking to each other. This is the first bottleneck that we oftentimes experience. Another one is that the medicinal chemists oftentimes have to wait for the comchems to get back to them. Obviously, computational chemists have a lot on their plate as well, and then come along the medicinal chemist with a question: hey, can you calculate that structure for me? Or, you know, what's the next idea that you can come up with? This is also stealing a lot of time from the pipeline. Then there are unsuccessful compounds synthesized, and we all know how expensive and frustrating it can be if there's a compound and this compound fails then in the success of the pipeline. Developing in the wrong direction can also be a bottleneck, that the wrong decisions are made at an early stage, and then much later you discover that this compound or the series of compound might be the wrong one for various reasons. Another one, and this is obviously something that we are dealing with a lot, is that software is overladen with unneeded features, and I will come to that point in a little while in more detail. Then there is something that we cannot deal with so much, but I wanted to mention it nonetheless, which is that a different direction is mandated from above. So management thinks that a certain project is not worthwhile pursuing anymore. So you have to start anew. This project is put in a drawer next to all the other ones that have been canceled, and you have to start from the beginning yet again. And this is sort of going into the same direction. This is shareholder pressure. I just want to mention these two here. Like I said, we can't do much about these, but I think we can address mostly all the other points above here with some points that I want to talk with you today in this webinar. So I believe we come to the first poll here. We have prepared a few polls. Christian?
Okay, so here's the poll. After we have introduced to you ourselves and also introduced the topic to you, we are obviously curious to hear and learn a little bit about your background, also to be able to adjust the presentation as best possible to your needs and interests. So please identify your personal background. And I see quite a few people have already given their clicks in the respective areas, and I'm going to close this poll in 5, 4, 3, 2, 1 now. And I'm going to show you the result. So roughly half of the audience is from the computational chemistry area, and then a third are medicinal chemists, and also a few biologists, crystallographers, and people not really fitting into any of these categories. Thanks so much for sharing this information with us.
Okay, yeah, let's continue then. So probably most of you will know this, but I wanted to mention it nonetheless. We have the pipeline here, and mostly what we are dealing with as software providers is people that we cater these software to is they're working in hit to lead and lead optimization. And this part of the cycle time is about 3 to 4 years long. So it's a substantial amount of time of the pipeline. And the costs that are eaten up in the hit to lead and lead optimization phase are about a quarter of the entire cost of 2 billion of a new molecular entity. So this is quite substantial, and obviously it's in all our interest to make this time as valuable as possible. So how can we do this? This is a little bit theoretical, but this is the pharmaceutical value equation. The value is proportional to the work in progress, the likelihood of technical success, and the value divided by the cost and the cycle time. And if you all remember your calculus, obviously this value will get larger if we reduce the costs and or the cycle time. So both ideally should be reduced in order to make the most of the pharmaceutical value equation here. This is the productivity P here. And I don't want to go into much detail about these here. These are the most obvious and probably the easiest ones to change. So this has also been a topic at the some of the Ivy League business schools, interestingly enough. And Professor Hughes once suggested that developing an evolutionary technology that improves the speed and or accuracy of an existing process, for example, developing a software for better information exchange within a large R&D facility. And he also went on with this, that it should be team-based, it should be an interdisciplinary technology. So I interpret this as, for example, interdisciplinary between the medicinal and the computational chemists. I found this rather interesting that this was also a topic at the Ivy League schools. So a few possible remedies for these bottlenecks that I discussed earlier can be, and again, I cherry-picked obviously to make this fit this webinar best, would be then, according to the slide that we had seen before, a tool that focuses on the ideation process, ideation meaning compound ideation of the chemists, with a reliable method to predict affinity, because affinity is one of the most important parameters that we have to deal with, not the only one, but one of the most important ones, that is visually communicated, and I will get into this in more detail later on, and is also able to predict ADME tox properties. These are, next to the affinity, other very valuable parameters that we can't lose out of our sight, because they have a fundamental effect how successful a compound will be later on in the process. That tool can or possibly be used by the medchems alone, not to exclude any computational chemist, but it should foster their communication with the comchems and take some workload of the comchems. So if the medicinal chemist can sort of boil up on their own a few ideas for compounds that they find interesting, they could then later on send those off to the comchems to have more calculations done and to validate those. But the first ideas, we think, should be made by the medicinal chemists. So I mentioned earlier the point of easy to use software, and I've incorporated an example here for a rather complicated word processor. And as you can see, there are so many more features and buttons that you actually need to write a text. This is obviously very professional software, but let's be honest, most of us, I would say 95%, are using some formatting and want to write mostly simply a text. Another word processor, on the other hand, is rather simply designed. Only the most necessary buttons are up here. You can write your text, and then you can open this format sidebar here and format a little bit. But it really has only the most important features necessary to concentrate on your writing. And this can be brought to the extreme with a few apps that are really simply focusing on the pure process of writing. So no buttons distract your work. Some of these tools allow you to write in screen-only mode and really have only a few formatting buttons here. So this has been a development that's been going on in the past years, and we sort of have taken this on when we designed our software SeeSAR, which you can see here a screenshot of. So you can see that we only have a few buttons up here in the menu bar. This is for loading a protein. This is for loading ligands. This is a preferences button. Meanwhile, this has changed a little bit. You'll see a more recent screenshot later on. This is your compound, visually communicating how well each atom contributes to the affinity. I will go into details later on. And this is a table where you can see the estimated affinity. And if you have more compounds, you'll also see an example for that later, how well they compare to each other or to a reference compound. Very important is this button down here, which deals with the torsions. So the torsions can also be visually communicated how they distribute in the CSD, the Cambridge Structural Database. So how often you see these kinds of torsions in small molecule, high quality crystal structures. Green is obviously very abundant. Yellow is they occur sometimes, and red, which I don't see here now in this example, would be you don't see this torsion at all. So coming to the calculation of the affinity, which is this really important parameter, is important to make this as precise as possible. And while we don't claim to be doing this on a quantitative scale, we can do this on a qualitative scale. And in order to do this reliably, we have engaged in a sort of a different approach here by focusing on the unsatisfied interactions that a compound makes with the protein. So, for example, this has been described by Lange in 2003. Unsatisfied hydrogen bonds lead to a 100-fold decrease in binding affinity. And this is a perfect example here. We have this compound, 7 hydrogen bonds with the receptor, and it binds with 3 nanomolar. If we omit this carboxylate group here, you see that all of a sudden 2 amino acids are unsatisfied. We lose 1 hydrogen bond. We only have 6 left, and we lose about 2 orders of magnitude in binding affinity. Now, this is an ideal case, but we have experienced this many times, and this is a very important concept, especially for scoring functions, to incorporate unmet interactions, because most other scoring functions, if not all, omit this very important aspect. So here is a more recent screenshot of SeeSAR. This is the screen that welcomes you when you open the software. You can download a PDB, you can open a project, you can load molecules, and I'll show you a live example later on. But let's first look at the sort of the machinery behind this. In order to calculate the affinity, we use a scoring function called hERG. And hERG is for hydrogen bonding and desolvation. Hydrogen bonding and desolvation. And this scoring function takes a very different view at energetics. It is based on physics only. So that means it can be applied to a variety of complexes. There's no calibration to any protein ligand complexes where you maybe can apply the scoring function only to a certain area of protein-ligand complexes. No, you can use this for protein-protein interactions, for ligand RNA, for ligand DNA, even for protein DNA and RNA interactions. So this is very widely applicable, and since it relates to the free energies, you can really compare this right away to any free energies that you have measured in the experiment. So I have to go a little bit more into the details here. If you are bored by any equations, you may check your email now. But how we calculate this is simply by comparing the unbound state with the bound state. We introduce the ligand from the unbound state when it is in the solvent and calculate for once the change in free energy of dehydration for each atom I, and then the change in free energy of hydrogen bonding for each atom I. And we simply sum all this up over all atoms I of the protein and the ligand, and this is our change in free energy that we are interested in. And this is a very simple equation. These two terms here can be made again out of these equations, which are very similar. We very consistently describe the dehydration, or in other words, the hydrophobic effect and the hydrogen bonds through the partial log p values for specific atom types. I will mention this on the next slide. There is a constant term here in front of the partial log p value, and then we have the change in Connolly surface for the dehydration and the change in number of hydrogen bonds between the ligand and the protein. And this is not incorporating any solvent molecule hydrogen bonds. So simply how many hydrogen bonds are formed between the ligand and the protein. And then the only difference that we have is the saturation factor down here. This is a temperature-dependent effect, how complete the hydrogen bond network in a solvent is at any given temperature. So for water at room temperature, where most experiments are recorded, it is 0.89 or 89% complete. And this is quite an elegant way to introduce the entropy here through the back door, because since it's a temperature-dependent effect, obviously this factor will change as well with the temperature. Okay, so how do we derive these p log p atom types for the log p? We used a small data set from the Hunch set, 500 organic small molecules with mostly one functional group, and we derived 3 atom types for nitrogen, 4 atom types for oxygen, and 1 for hydrophobic. And as you can see here in this example, when we dehydrate, for example, an sp2 oxygen, this is an energetically unfavorable process because the p log p value is negative, and if we plug this into the equation we've seen before, the equation becomes positive because we have a negative constant factor. So this is energetically unfavorable, whereas obviously dehydrating an aromatic carbon is an energetically favorable process. This is also called the hydrophobic effect, as everyone knows. And with this rather simple concept, with those p log p increments, the solvent accessible surface, the saturation factor, and the number of hydrogen bonds, we can correctly predict, for example, the hydrophobic effect or the quenching of a hydrogen bond from vacuum to water, or the affinity loss due to an unsatisfied hydrogen bond. And this is most important for our affinity calculation to give the compounds the right order in which you should be looking at them. So like I said, HYDE is dealing only with hydrogen bonding and desolvation. I have an example here, 1-GKC. You can again see the quality of the torsions here for this compound. And if we look at the contributions for each atom, you see how visually communicative this tool is, because you can immediately see without looking at any numbers that this atom here is giving you a bad penalty for your affinity. This is giving you a smaller penalty, and this is really contributing the most out of this entire molecule because it is forming a salt bridge here to the metal. Now, this atom pays the penalty, or has to pay the penalty, because there is no hydrogen bond formed between the carboxylate of the backbone here, the carbonyl of the backbone and the NH. And therefore, we have a large desolvation penalty, which is not overcompensated by any hydrogen bond energy. And this is why we have this penalty, and this is why we very likely lose about 2 orders of magnitude in binding affinity because of this missing hydrogen bond here. In other words, if we were to repair this, either replace this with something that would form a hydrogen bond, or at least replace it with something more hydrophobic, we would gain a binding affinity. Okay, so we can also put some more numerical flesh to the bone here. If we label these, you can see here that the penalty we pay is about 10 kilojoules per mole. And this is basically divided into a desolvation penalty for the ligand and the receptor. The ligand pays a much larger penalty. That's why we would gain about 7 kilojoules if we were to replace this with something more hydrophobic. If there was a hydrogen bond, we would see this in the interaction here, and if this value was larger than that and that, respectively, obviously we would have a small gain. On the other hand, we have this oxygen here. We have a positive contribution, or a favorable contribution to the binding affinity of -11. And as you can see, the interaction on the receptor side is very large, and the desolvation penalty for both the receptor and the ligand is overcompensated by the interaction energy between the oxygen and the metal ion. Okay, so I guess we have a next poll here, Christian.
Mm-hmm. Okay, so the next question now is in reference to what you have seen so far, a tool like SeeSAR for detailed analysis, understanding the situation exactly, and leading you to a very rational design approach. Is that like your day-to-day use tool, or do you rather use other means to arrive at similar conclusions, or do you not consider this a necessity at all, something like that? So I see answers to this poll are coming in. Please give us your insights and your opinion on this question. A few more coming. I'm going to close the poll in 5, 4, 3, 2, 1 seconds now. Okay, thanks again for the responses, and here you see the response for yourself. The vast majority is saying, yeah, this is very important. Analysis like this I do all the time, but a sizable fraction still considers there a gap or an open need still to be addressed, and maybe we can get to more of that in the next part of the presentation.
Okay, let's continue then. Can everyone see the slide? Can we see the slide again? Christian? Oh, you unmuted yourself already.
Yes.
Okay, cool. All right. Yeah, okay, so I
muted.
Hydrogen bonding and desolvation. And for that reason, we need to have very good crystal structures in order to get these hydrogen bond geometries and networks correct. There is, however, a slight problem there, that the PDB files that we are dealing with are mostly, or very often, I should say, not as good as we think they are. And Derek Lowe had this wonderful quote here: "We tend to believe PDB files are a message from God." And unfortunately, this is already is always a little bit in the neglected, I should say, that the PDB structures that we are working with are really not ideal. So we, when we implemented hERG, we dealt a lot with crystal structures, and we had a subset of the PDB bind, which was called a high quality, in quotation marks, data set of the PDB bind 2007. And we looked at all those 200 complexes, almost 200 complexes, by consulting the electron density maps. And it turned out that more than 50% had structural deficiencies. So, for example, there was missing electron density here, so the ligand was kind of arbitrarily placed in the active site. Same with the alternative conformations, very flexible ligands. They had very poor density, as you can see here, and this ligand can really be anywhere in the active site. There are high temperature factors in contact with symmetry-related subunits. So all this is rather disappointing and frustrating when we deal with this, because if we use these complexes without further analysis or correction, we might think that the scoring is very inaccurate. So, for example, here, the 2F-ZC complex, the experimental binding affinity is -15, but the calculated one is even positive. So we might come to the conclusion that the affinity calculation is not working. However, if we look at the electron density, we see that it's pretty poor here for the ligand as well as for the binding site. And this is really responsible for kind of messing up the affinity calculation here. We see this in this coloring, again, which is visually communicated by the tool. It colors the spots on the protein surface where there is penalties in red. So there's a lot of red stuff going on here, and likewise on the ligand, which you don't even need to look at the values here. All right, so next example, this is also a crystal structure where we would expect a hydrogen bond between this aromatic oxygen and the nitrogen of the receptor. The calculated affinity is rather low, but we see that this penalty is really bothering us. So what happens if we dock it? This ligand is docked back into the pocket. It now has a green atom, which means this is favorable. All the other atoms are pretty much the same. And we got an improvement here by twofold. And the RMSD is still very much in the same area as it was before. So again, this might be a flaw in the crystal structure that we can correct, but that also the tool, the software, communicates to us. One last example here is this pyridine ring where the nitrogen is facing towards the protein and the aromatic carbon is facing towards the solvent, which might not be the case. It might be the other way around. And you can see here by the estimated affinity that we are in the millimolar range, although we know that the binding affinity for this particular ligand is rather in the nanomolar range. So heavily underestimated, and again, we might say, you know, this is not working very well. But if we turn this around, we see that the carbon is much happier in the protein, and the nitrogen is much less unhappy when it's pointing towards the solvent. It would ideally like to be hydrogen bonded somehow, but it's at least less unhappy here, and we can see that the affinity is indeed moving into the right direction. There's a second spot on this in this example where an atom is unhappy. This nitrogen here is very unhappy because it's missing a hydrogen bond towards the carboxylate. You can see that there's no interaction energy, a large unhappiness on the ligand side. So if we correct the protonation state here, we do see that all of a sudden we do have a large contribution for the interaction energy, which overcompensates the desolvation penalty. And now we do have a very small but noticeable contribution from this interaction here. So at least the unhappiness is gone, and this atom is now sort of neutral. So the reason why this is important is that it is it can be regarded as a weakness, but I would rather claim that it's actually a strength of the scoring function here that is so picky with respect to these interactions. Because if we look at the hydrogen bond potential here for an NHO hydrogen bond, we do see that as soon as we move away from the optimal distance or angle of the HO here, we will climb up the potential wall very, very quickly. And this is exactly what we see also with our scoring function in SeeSAR. And in order to avoid that happening, we need a good correction to optimize the hydrogen bond network and the position of the ligand in the active site. And this is shown by this example here. We have, again, an example from the PDB. No correction done, simply scored. And again, we might say, well, this is not working very well. But if we correct the hydrogen bond network, we see that all of a sudden the NH3 here is very happy when we did this flip of the amino acid here. Some other atoms are also more happy, and we gain about threefold in binding affinity. And if we now correct for the position, we basically wiggle the ligand a little bit so that all the hydrogen bond angles and distances are optimized. We gain another 5 kilojoules in binding affinity. So this is a crucial aspect of the scoring, which has to be done a priori before we score. But as you will see in the live example later on, it's a really quick process, but it's a necessary process in order to get all these geometries right. So here's an example for the cost of a hydrogen bond, how important it is to have really all hydrogen bonds in place. This is to BRB. The oxygen here is opposing a carbonyl oxygen on the protein. And we can see that both the experimental and the calculated affinities they're okay, but they're not great. So what happens if we replace this oxygen with a nitrogen? You can see we have a nitrogen here. The nitrogen is forming now a hydrogen bond with this carbonyl, and we gain 10 kilojoules in binding affinity, which is exactly what I told you in the beginning with this example where we remove the carboxylate group. So again, 2 orders of magnitude in binding affinity, 1 by this very simple correction. So this was sort of concerning the theory and the background, why it is that we need this affinity calculation in the way that we're doing this. Now let's tell you a little bit how this can make the drug discovery process more efficient. You can see here, this is a magic methyl in this ligand. And simply by comparing these two, and actually without looking at the numbers, you can see that this corona here, this affinity corona, is the largest on the entire ligand. So this methyl group is missing here, and that's why we have a rather bad inhibition constant, which changes if we introduce this methyl group. There are other groups in this publication that we also tried out with our software, and they behave mostly similar to what we see in the experiment, all in the same ballpark with the exception of fluorine maybe. But the software is able to at least distinguish between the most important ones here. That's very good. Another effect that I haven't shown here is that the boost in affinity mostly comes from locking these two phenyl, the pyridine and the phenyl ring, into a very favorable conformation if the methyl group is in ortho position, which brings most of the boost in affinity. And we can make this visible also if we look at the torsions in the software. The torsion with the methyl group is actually yellow, whereas if you remove the methyl group, the torsion turns red. So also a little bit of a hint that introducing this methyl group is a very good idea. Then again, lead optimization. Is it a good idea, for example, to replace this methyl group here with a phenyl ring? Yes, it is. The software tells you that this is here the right-hand structure. The affinity is moving into the right direction. Actually, it's the left direction, but it's the correct direction, meaning towards nanomolar. You can see this in the experiment. This is the experimental values as well. There's a boost in affinity. So, telling you really quickly, this is a good idea to make this change. And you can do this all in 3D in the active site, and it takes you maybe 5 minutes to determine whether this compound is better or this compound might be better. We also have the possibility to select different binding modes. So this tRNA guanine transglycosylase has 2 binding modes because the binding site is rather large, and if you see those two in 2D next to each other, there's really no way of telling which one is correct because you have 4 hydrogen bonds for both of them. But if you look at the structure in connection with the coloring of the active site, you can immediately see that this cannot be the right one because there's so many red warnings here on the protein surface because actually a lot of hydrophilic atoms are blocked off by the ligand. So this must be the correct one. And if we now compare this with the numbers, this is also in very good agreement with the experimental value here of minus 23. And the RMSD also tells us that only the left side can be the correct one. So then another example for compound prioritization. Sometimes you have, or not sometimes, most of the time you have a lot of compounds and you would like to decide, whether that little change or that little change is good, and you should make this compound. And this has been done here for this thrombin example. All these compounds only vary in the S1 occupant, so the changes are rather small. All this has been left alone, and you can see the different variants of the compounds here. And again, without looking much at the numbers, you can see that this is the best compound because there's no red warnings on it. By the way, don't get confused. This is the older visual communication. We hadn't introduced the affinity coronas at the time, but the message is the same. This is all green, so this is the best one. And this coloring scheme actually helps you very well also doing analysis. Why is my compound not performing so well? And I'm going to take this example here, that the introduction of this chlorophenyl ring here is better than having a fluorophenyl, but it's actually not ideal. The reason for this is, on the one hand, we have an unsatisfied glycine here, which maps its unhappiness onto the aromatic carbon. The aromatic carbon as such is happy, but the protein is not. The chlorine is a good idea because we have the hydrophobic effect in this part of the pocket. However, there is a big unhappiness on the nitrogen here. And why is this? Well, we think that because we have this chlorophenyl ring, the ring is pulled into this hydrophobic pocket, pulling this ideal angle apart so that the hydrogen bond angle is not ideal anymore. You can see this here. It's 49 degrees, which is really ways away from the optimal hydrogen bond angle. And this is the reason why this gets this large penalty. So, this is an effect that's maybe not very obvious if you didn't have these colorings on your molecule. So, if you remember this structure beforehand, you can see that the group they have introduced is much more bulky. So this pulling effect into the hydrophobic pocket is much less expressed. Okay, so here comes a live example, and I'll see if I can get this over to you. Let's see. I'm going to start up SeeSAR. Can't see anything yet. So here's SeeSAR. You have seen this screenshot. Actually, let me make this a little bigger. You've seen the screenshot, so what you can choose to do, either download a PDB file or open a project file or a PDB, or simply load molecules, because you can also work on molecules alone without the protein. So the structure that we have been looking at previously is 2ZC9. This is the thrombin structure. This is pulled from the PDB website, and initially you see the protein and its 3D structure here. You see the co-crystallized ligand, you see all the water molecules, and you see the estimated affinity of this compound in the table next to a bunch of other stuff that I'll go into detail in a second. So now what you can do is, for example, add some ligands that have been looked at in the experiment already. I've prepared this here. These are about 16 molecules that are now live calculated with respect to their affinity. So the first step, as you remember, is optimizing the ligand and the hydrogen bond network in the protein, the position of the molecule, and then it is scored. And the score is what you see here. We also calculate the ligand lipophilic efficiency, the ligand efficiency, and we give, just as with the affinity, just a qualitative value. Because like I said in the beginning, what we are after is not a quantitative description of the affinity, because this is impossible to do, as everyone might know. But in a qualitative sense, we think it's pretty much possible, because what you're after is simply a comparison between your reference state, and this might be the ligand that's co-crystallized with your protein, and the ligands that you have designed in your head, or you have been docking into the structure or whatnot. So this is really a more relative way of addressing the affinity here. So this is again the protein here. You can click on the table, you'll see a more detailed view of your ligand. Again, here's the this nitrogen that's sort of pulled apart by the chlorine in this pocket. It's upside down now, so in the picture before we've seen it up here, but now it's down here. Here's the chlorine. You can add labels to this to gain some notion how much this atom is contributing or how much this atom is penalized. So this is the co-crystallized one. We can sort this according to affinity. So this would be the best one here. And if I'm not mistaken, yeah, this is the one that we also saw on the slide, the one with the bulky group here that basically prevents this nitrogen from being too much away from the optimal hydrogen bond angle. So you see that the nitrogen here is green rather than red, and there's no red atoms on this in this structure. So what's new now is that we can expand this table, and we have the possibility to calculate ADME tox properties, and those are the ones that come from the StarDrop software from the company Optibrium. We have recently engaged in a collaborative partnership, and we have incorporated their ADME tox property calculation into SeeSAR, so you have way more than just these to calculate. So, for example, some CYP450, a blood-brain barrier, or hERG values, and also a solubility estimation you have here. So these can also be sorted according to these parameters. So you have an idea how these compounds behave according to these important values. And also, if you have properties in the SDF file, you can enter these into the table. So, for example, the exact molecular weight, well, we don't really need that, but the affinity, the experimental affinity would be a nice thing to have. And this is in this column now. Again, you can sort according to this and compare it to the calculated one. All right, this is the most important features. There will be another webinar in March, I think, which deals a lot more with how do I use SeeSAR for my projects, what functionalities are there, how can I use this most efficiently, and this will be more of a hands-on. This was sort of the first part with all the theory and some examples. So stay tuned. In March, we will have sort of the second part of this, which will deal more with an applied examples, and you can download SeeSAR before the webinar and then sort of follow follow us what we are doing here on screen. Okay, so let's get back to the presentation here. One last example I wanted to show you is more of a computational example, which might be interesting for the computational folks in the audience. There was a screening effort at Bayer. Actually, Bayer is the cerebral home of the scoring function height, which is in SeeSAR now. So Gudrun Lange is the conceptual head behind this. And they used a preliminary version of height, sort of a prototype, to do a virtual screen. And they submitted their virtual library of 4.5 million compounds to a FlexX docking. FlexX was docked, was used to dock these compounds into the active site of a polymerase. Then about 2,200 molecules get a height score below minus 27 kilojoules. So this was sort of a cutoff for, is it a good binder or not? This value has changed a little bit over the years, but essentially it's still in that ballpark. And they were, so these 2,200 molecules were inspected concerning the binding mode by the researchers at Bayer. And they found that 974 of these had a reasonable binding mode. Of these 974, 172 compounds were available at vendor catalogs or were already purchased and on the shelves. And they were tested in affinity assays. And it was a very good success that 116 of these 172 were actually confirmed hits in several classes and plant active compounds. So that converts to an almost 70% hit rate. So obviously you can imagine that this was a very good result, a very great success for this rather preliminary version of the scoring function that we now have in SeeSAR. And it has been only improved since. So you can try this your own in your projects and on your examples. So this brings us, I think, to our last poll here. We ask you a little bit for your help in the further design of the software that we introduced to you today. And please let us know which feature you would cherish most in a software tool. And I think this time you can select multiple answers here. Christian?
Yes, I already started the poll. Question being, what functionalities would you like to see with highest priority be realized in SeeSAR? You may have noted in the past that SeeSAR is updated almost on a monthly basis. We add new features and very much listen to the feature requests from users. So this is not just for the fun of asking, but really has an influence on our priorities in the design of the next stages of SeeSAR.
Yeah, and therefore maybe if we can ask you to maybe limit yourselves to two and a maximum of three. It obviously doesn't make sense if you check all of them, because then it's not really a prioritization. So if there's one or two and at the most three features that you would like to see or that you think may make your efforts more efficient, then tick those, and we will use this as information in our further development.
Yeah, quite a few have already responded. I'm going to close the poll in 5, 4, 3, 2, 1 seconds now. And here is the result. As you can see, yeah, it pretty much spreads across the field. There's a slight advantage in favoring rating the happiness of water. I think that's a very good one. Yeah, getting to know which water to replace would be potentially a good idea. Certainly is a big point in the design. But this is closely followed by some of the others. So thanks very much again for the response, and let's continue with the rest of the presentation now.
Yeah, there's basically just one slide, namely the summary side. And so what we showed you today was a software that's mainly dedicated to the medicinal chemists, but not to exclude any computational chemists, but to make them more independent and to hopefully further some of the ideas that the medchems have in their heads. As I said in the beginning, we have experienced that the communication between both is sometimes a little, how should I say, obstructed. And the medchems oftentimes have their own ideas of what compounds they want to make or what compounds they are interested in. So I think if we let them do this and then follow up these compounds by the computational chemist, I think this would be beneficial for both of them. It would take some workload of the computational chemists. It would be, I think, a more focused approach on really the compounds that matter. And this software really helps in this compound ideation process. What's also important to note is that obviously the affinity calculation is very important, and we are not after a quantitative calculation here because this is, we think, impossible. But it is very possible to do a relative comparison between different compounds and to see whether the change in a compound that you've made in the protein in 3D very interactively is beneficial to the binding affinity. So you can really optimize and you can generate tens and 20s and 30s and maybe 100 of different ideas a day, and then at the end of the day sort them and see which ones are likely to be most promising to further pursue in relation also to the relevant ADME tox properties. So if you see a compound that has good affinity but that has other bad ADME tox values, you might want to stay away from this. So yeah, this software obviously is meant to be tried out, and every one of you guys that participated will get a free license for a certain amount of time. So you can try it out yourself. We are, of course, happy if you share your feedback that you had with the software with us. This little poll we had at the end shouldn't be the end of it. So if you have a good suggestion for a use case, something like, hey, I want to do this and that, and in order to do that, I need to accomplish this and that, and this is valuable information for us for the further development. And of course, we want to stay close to you guys to really portray every sort of use that you have for the software and to make the work more efficient for you. So in that sense, this is the end of the presentation, and I guess I'm hoping we have quite a few questions.
Yeah, we have a few questions. I marked a few as being best answered at the end of the presentation. Can you see the short list, or should I read the questions and you answer them, Carsten?
I can't see the short list right now, but let me see.
Let me then just ask the question, and you can provide an answer. Okay. Okay. So the first one is when calculating the energy, the height energy, desolvation and dehydration. Dehydration and hydrogen bonding part. Are the water molecules in the vicinity considered during these calculations, and are the waters then implicit or explicit?
Yeah, so the question is yes and no. So the waters are considered. So for the dehydration of the ligand and the protein, we only consider the solvent accessible surface. And obviously it doesn't make much sense to calculate the solvent accessible surface for any water molecule. However, if the ligand is forming a hydrogen bond with that particular water molecule, then it adds to the affinity, but only if it's a conserved water molecule. So SeeSAR keeps water molecules that have at least 3 interactions with the protein and or ligand, and those are considered to be conserved, and those could then potentially hydrogen bond to a ligand or any development that you make on the ligand.
Okay. Next question is, can SeeSAR be used to optimize the ligand conformation and orientation, or does this need to be done externally before analyzing with SeeSAR? And I already provided a brief answer to this, so maybe I can take this one.
Yeah.
And give a little bit more feedback. You can do quote-unquote docking or post-generation, however you want to name it, with SeeSAR. That means taking the compound, placing it in the active site, and then followed by the SeeSAR analysis. And given a position of a molecule in the active site, SeeSAR anyway always optimizes the conformation and position of the molecule, but this is only a local minimization. So it really depends a little bit on your expectation. If you're providing SeeSAR with a starting point, a local minimization including conformation and position of the molecule is done. If you are starting from scratch, yes, you can do this with a docking functionality in SeeSAR as well, but you can also do the docking outside. All right, next question then is it possible to optimize ligands with SeeSAR? Is it only possible to optimize ligands with SeeSAR on the basis of crystal structures?
Yeah, this is a very good question, and obviously we all, many times we come into the conundrum of what should we tell our customers, which structures to use. So as I have tried to explain today, is that we need to have very, very good crystal structures in order to get those hydrogen bond networks correct and the optimal hydrogen bond distance and angle in order to get this overcompensation of the desolvation penalty. This doesn't prevent you from using SeeSAR on, for example, homology models, but I would be highly suspicious of the results that I'm getting. If a homology model is the only thing that you have, then this might be your best guess. But again, I would be very wary of the results that I'm getting. So the careful selection of a reference state and careful observation of things that happen during and after the calculation is very necessary. There have been cases where I've said, or where we've said, rather not use it on homology models, for example. NMR structures are similar. They are usually very flexible and highly. The motion is all over the place. So that's the same thing. How reliable is a hydrogen bond that's formed, with very movable parts of the macromolecule. So again, if that's not in a region of the protein or the RNA that's not so mobilized, I would be very careful. So the ideal case is, yes, I use it on high quality crystal structures, but if you don't have one, then it's up to you if you want to do that.
Okay, so I think we have to hurry up a little bit. People, I guess, have allocated the 1 hour and start to drop out. We want to cover the most important questions here. of course, for all of you. The next one is, in the live demo, does SeeSAR add hydrogens and or correct flaws in the PDB, like missing side chains or missing positions in the PDB or something like that, or at least give warnings?
Yeah, that's a good question, and I kind of omitted that point a little bit for the sake of time. So it's again a yes and no answer. Yes, it does add hydrogens, and it does correct for hydrogen rotors in order to get the hydrogen bond network right. But no, it does not correct other flaws in the PDB. So it doesn't add any missing side chains, and it doesn't even give you a warning. It only gives you a warning sort of as a side note, because if there's a side chain missing that should be there in order to form a hydrogen bond, you will see a red affinity corona on the ligand and or the protein. So like this one example that I showed where the crystal structure had those many flaws, this is kind of the way that SeeSAR tells you, hey, there's something going on.
Okay, next one right up there. Are there plotting features in SeeSAR, for example, predicted versus experimental data?
Yes, I didn't show this, and this will come up in the next part in March. You can compare the experimental and the calculated values, which we have kind of hidden a little bit because there's not much value in there. We think to make sort of this correlation analysis, but in order to cater to those people who are interested in it anyway, this feature is there, and it's maybe I can show this very briefly. Let's see here. You will find this down here in this little performance check tab. You can select which ones to include in the performance check and which ones to omit. But again, like I said, this will be a part of the next the next webinar after the next one.
Okay. So next to last question: is it possible to make an alignment of several ligands in the active site?
No. That's not possible. So if you are after an alignment, you would have to do this with an external tool such as FlexS or any other alignment tool. You can load different ligands into the active site, just as I've done here, and you can sort of select a reference state and then compare different conformations of ligands. But the alignment calculation as such is not possible right now.
Okay, and the last one, I think an important one, because there seems to be a misunderstanding. I'm reading here, the PDB file does not have any hydrogens due to the known issues with hydrogens in crystal structures. But at any rate, if we have to add hydrogens ourselves, why is it important to start with a crystal structure or homology in the first place? I guess we should clarify here that you don't have to do the hydrogen assignment in protein structures yourself or outside of SeeSAR. Actually, this is one of the very good algorithmic solutions that we have implemented in SeeSAR, that all of this is done automatically and with an optimization function that really has the overall best hydrogen bonding network as a goal. So I think we have a very good solution to that, and I think it's a misunderstanding if we conveyed that you have to do this outside of SeeSAR.
Yes.
Okay. All right. Yeah. To all the others, I hope we have covered your questions. Some of them you're hit on the same topic. I hope we addressed that reasonably well. Otherwise, of course, feel free to to contact our support via email, and we go at full length detail to answer all your questions. And Carsten, last pointer to the next meeting once more.
Yes, the next meeting will be, hang on a second, I'm gonna start the thing here. The next meeting again is February 23rd, 2016 at 10 a.m. Eastern Standard Time. That is 4 p.m. in Europe, a little earlier here on the West Coast. But that will deal with the de novo design in synthetically accessible compound universe. So we'll invite you all to participate again. Again, it's free of charge. Sign up the usual way that you've done before, and then we will tell you about how to make most use of the compounds you have on the shelves, and what you can do with these virtual compounds that are out there in space, and how you can access those.
Okay, thanks once more for the live participation, giving us all this feedback in the polls, and also here doing the question and answer session. And thanks, Carsten, for an excellent presentation. Thanks for all of you staying with us and participating in this webinar. Hopefully see you again in about a month's time for the next webinar. Bye-bye.
All right. Thank you. Bye-bye.